Welcome back, guys. In this set of video lectures, we'll be talking about the DNA replication process in eukaryotes. Okay, we have seen the DNA replication process in prokaryotes, especially in E. coli, but in this case, we'll be seeing the process in eukaryotic cell. Now, the replication process in eukaryote is much more complicated than prokaryotes, because in eukaryotic DNA replication, it involves many different set of proteins, protein complexes to ultimately deal with the situation. We need the major enzyme, that is the DNA polymerase, that is doing the replication process, is far more complicated in eukaryotic case compared with the prokaryote one. Okay. So in this first video, I'll be going for 4 different video lectures regarding this DNA replication process in eukaryotes. In this first lecture, I'll be talking about the basics of the DNA replication process. Now, overview of DNA replication process in eukaryotes and type of enzymes involved, and also we'll be talking about where exactly this eukaryotic DNA replication differs from the DNA replication in prokaryotes. Okay. And from the next 3 lectures, we'll be talking about the initiation, elongation and termination of DNA replication process in eukaryotes. Okay. So in this video, we are majorly concerned about the enzymes that are used in DNA replication and the basics of it. Like any mode of replication, like in prokaryotes, any case, the DNA replication process have certain kind of rules, right? The rule is the idea of replication. That means we have a double-stranded DNA, say we have this double-stranded DNA, let me draw it with 2 different colors, and both of the strands separates from each other, okay, and we use both of the strands as template to produce a new daughter strand, right? That is the idea of DNA replication all the way around in prokaryotes as well as in eukaryotes. So those strands are called parental. Now we add the new strands by using one strand as a template. So it will produce 2 strands, 2 double-stranded DNA like this. And let's say these red strands, these are the daughter DNA strands, and these are the parental DNA strand. Okay, that's how the new DNA strands are produced. This is the idea. But during this production process of synthesis of new strands using old strand as a template, this model is called as a semi-conservative model of DNA replication, and true for all DNA replication in prokaryotes as well as in eukaryotes. Now, during this process, there is a directionality of this replication. So, for synthesizing a new strand of DNA, we need to separate the old strands from each other, right? So, in any DNA replication, it's very important to separate the parental strand first. So, we do that. We need some enzyme to separate the parental strands from each other, okay? And usually the DNA never stays like this, it is helical and it is coiled, right? For that reason, we need to unwind the DNA, then separate the strands. So this process of unwinding the DNA and separation of strands are brought about by specific enzymes called helicase, DNA helicase. Helicase is ATP driven machine enzyme which takes energy from ATP hydrolysis and separates the DNA strand from each other. But in case of prokaryotes, simple helicase, one helicase protein can do this task. But in eukaryotes, multiple proteins are required instead of one. Usually 3 proteins working together to separate the strands and to form a structure what we call a replication bubble. Okay. What is the bubble? We will be seeing later. But we need multiple proteins there. Okay. 3 different proteins at least. CTD, CDT, then CDC45 as well as MCM. So all these proteins working together there. But in this case, the separation is required. Once the separation is done, then the second stage is priming. In any case, the major enzyme for the polymerization of DNA, because the DNA replication means we are synthesizing new strand of DNA. So we are making nucleotide polymer, right? Deoxyribonucleotide polymer is called DNA. So for the polymerization, we need an enzyme called DNA polymerase. In prokaryotes, we have specific DNA polymerase called DNA polymerase 3 for the whole job. Okay. But in case of eukaryotes, we have other type of polymerase like polymerase delta, polymerase epsilon, polymerase alpha. These are the type of polymerase we find in case of eukaryotes. But in case of prokaryotes, we have polymerase 3 for major process and polymerase 1 for small synthesis of small fragments after the process is done of removing the priming part. This is the major thing. But remember, this polymerase enzyme which we have talked, none of these DNA polymerase enzymes can start synthesize on its own. They cannot start or initiate the replication process. They need a free 3' hydroxyl to start the replication process. They need this free 3' hydroxyl to initiate the replication. For that reason, this 3' hydroxyl is required for all of these different types of polymerases. So whatever, once you get this 3' hydroxyl, then they will initiate attaching nucleotide sequences one after another and start stretching it, but cannot initiate the first one. So to have this first 3' hydroxyl, it requires a stretch of nucleotide sequence, and that stretch of nucleotide sequence is called as a primer, and this primer is synthesized by DNA dependent RNA polymerase enzyme, because RNA polymerase on the other hand is very versatile. RNA polymerase on its own can initiate adding nucleotide sequences. So that's why we need RNA polymerase to add some ribonucleotide sequences at the very beginning, which will provide the free 3' hydroxyl to elongate by the DNA polymerase. So the stretch of 20 to 25 nucleotide sequences made by RNA polymerase, that is called RNA primase there, because the enzyme is called primase. Primase add these nucleotide sequences at the beginning so that the DNA polymerase can elongate that. Okay. Now, where exactly they elongate? I mean, which direction they exactly elongate? If we see this one of these cases, where exactly it will elongate and which is the directionality in this case? The answer in all this case of DNA replication in prokaryotes as well as eukaryotes, this directionality is 5' to 3'. Always the directionality will be 5' to 3' for the newly synthesizing strand. For example, let's say in this case, if this is the 5' to 3', the synthesis direction will be this. So the opposite template strand is this, and in this one is this. So synthesis process will be this. So see, if we look at these two different parental strands, and if we are imagining to produce the new strands, one will synthesize towards this direction, another will synthesize towards this direction, opposite direction. But the interesting idea is that in prokaryotes, the enzyme DNA polymerase 3 is the major enzyme for the polymerization event, for the elongation event and adding nucleotide sequences to produce DNA in both the strands simultaneously. It works to add nucleotide sequences in both the strands simultaneously. But as we see, the directionality is opposite, and there is two different subunit of polymerase 3 attached by a single protein unit called tau unit in prokaryotes. But still it is holding both the strands together in opposite orientation. So how could they do this? And the answer for that, the structure. During the preparation of new strand, they form a loop-like structure and make the directionality of both strands at the same way, at the same direction, so that the process can occur simultaneously as the DNA polymerase will move from 5' to 3'. Okay. But remember, for this particular reason, one strand is denoted as a leading strand which synthesizes almost without any interruption. Without any interruption and disruption of synthesis, nucleotide sequences are added one after another. It keeps synthesized. Let's say this one. This is the leading strand. This is considered as the leading strand. On the other hand, the other strand synthesis will have disruption because they form the loop. The strand where they form the loop has the disruption. It will not produce long stretch of nucleotide sequences at a time. It can only produce small stretch of sequence at a time, and the polymerase will fall off. Okay, so this is a problem here. So for this reason, the other strand, we call it a lagging strand because the synthesis is discontinuous. We call this a lagging strand. So the problem usually arises with the lagging strand synthesis. There is no problem in the leading strand synthesis because it works fine without any interruption. But in lagging strand, we have small stretch of nucleotide sequences and DNA polymerase falls. So just imagine, so if multiple times DNA polymerase can add small stretch of sequence, so again if it needs to start adding nucleotide, it needs again another primer. So multiple primers are required in the lagging strand during the synthesis, while there is only one primer required in the leading strand synthesis. And this is true for DNA replication process in prokaryotes as well as in eukaryotes. Okay, in both this case this is true. So multiple primer required in lagging strand synthesis, and they synthesize only small fragments of nucleotide sequences at a time. That is called as a Okazaki fragments. Okazaki fragments, based on the discoverer of the process, discoverer of these fragments. Okazaki fragments are very smaller in case of prokaryotes, very smaller, 20 to 25, 30 base pairs. On the other hand, in eukaryotes, the Okazaki fragments are larger. They are pretty big Okazaki fragments. 200, 300, 400 base pairs long Okazaki fragments are formed in eukaryotes because the eukaryotic genes are larger, long. They need to produce a lot of, they need to polymerize a lot of DNA compared with the prokaryotes. Okay, there is a difference. So this is the basic enzyme that we require. The difference mainly in between the type of polymerase we deal with and the way polymerase interact and produce the strand. In prokaryotes, we have polymerase 3 mainly doing all the polymerization stuff, and polymerase 1 does the job of removal of the RNA primer and filling them with the deoxynucleotide sequence, because the primer is ribonucleotide sequence. So we produce a kind of ribonucleotide and deoxyribonucleotide hybrid. We don't need that. So these primer components are removed and then it is replaced with deoxyribonucleotides by the DNA polymerase 1 in prokaryotes. While in eukaryotes, the process of replication is controlled by 3 different enzymes and all together, like the polymerase alpha, polymerase delta, polymerase epsilon. All of these 3. Usually polymerase epsilon is taken for the synthesis of leading strand, polymerase delta is for the synthesis of lagging strand, and polymerase alpha is required for the initiation of polymerization. Even after the primer is there, in eukaryotes, there is a short stretch of nucleotide sequences produced. 2 to 3 or 4 nucleotide stretches are produced by this polymerase alpha, and that is required. So polymerase alpha in eukaryotes is called as the initiator polymerase to start the polymerization process. Okay, and rest of the polymerases are working as a full-time polymerase to start adding nucleotide sequences to produce the DNA for a lagging and leading strand. So this is different for both of them. Another thing is different is the region where this process takes place. In prokaryotes, the polymerization process takes place in cytosol because prokaryotes do not have nucleus, right? So everything in cytosol. DNA replication also takes place in cytosol, and it takes place more than once. Okay, and once it takes place once, the cell divides, splits into 2 different cells. Okay, there is no other different stages and phases of their process. While in eukaryotes, if you look, it is much more complicated, and in eukaryotes the DNA duplication process occurs only once in a cell's life cycle, right? Then the cell will divide and then transfer the other copy of DNA to the daughter cell, right? And usually it takes place during the S phase of the interphase, because if you look at the cell cycle phases, interphase and M phase, in interphase there is G1, S, G2, three different phases. The process, the enzyme buildup start to occur in the G1 phase, and then this DNA is prepared for opening and start replicating in the G1 phase. Then in S phase they start this elongation process of DNA synthesis, and at the end of S phase the whole process is done and we have two different copies of the same genome. So it occurs in the S phase of cell cycle. Okay, that is about the eukaryote system. And the final thing is in prokaryotes. If you look at in prokaryotes, the genome is small, simple, right? There is a particular origin of replication called ori that is present in prokaryotes. They start the process from their ori and they'll start doing this, then the whole process is done. But in case of eukaryotes, the genome is linear. In some cases of prokaryotes, the genome is circular. So it's very easy. Different approach is taken. But in case of linear DNA in eukaryotes, there are multiple ori or origin of replication units to start. Okay, and then they will merge with each other. Another thing is that in eukaryotes, as this is a linear DNA, there is an end replication problem, because each time the replication is done and DNA is producing new DNA strand, the end part of the template DNA is not taken as a template, because the primer, the RNA primase, primase enzyme, unable to sit to the terminal region, and as a result the elongation process is not possible at the terminal region of the template. As a result, each time the replication occurs and replication cycle passes, the telomere section, telomere means the terminal section of the chromosome, start to shrink in eukaryotes, and that's called the telomere problem, and also it's called as the telomere shrinkage, as well as it's called the end replication problem in eukaryotes. And this end replication problem in eukaryotes is treated with another enzyme called telomerase. Telomerase enzyme. This telomerase enzyme helps to figure out this end replication problem, so that its telomerase actually add multiple nucleotide sequence at the end as a template, so that even if they get shrinked, still the actual genetic material content remains there. It's not getting cleaved. So that is the idea of telomerase there. It's a template carrying polymerase type of enzyme which adds its own template and start adding the nucleotide sequence on its own, right? So it's a versatile enzyme there. So this is completely unique in case of eukaryotes compared with the prokaryotes. So in a sense, this is the DNA replication process comparison as well as the basics in eukaryotes. So we will see three different stages in eukaryotes. We will see the recognition of the origin of replication, that is called ORI site. ORI site, that is the ORI site for the origin site for the DNA replication to begin. And there are a set of proteins, right, called the replication recognition site or recognition protein. So that complete recognition protein complex will form to that ORI site, and then they will bring more and more proteins on that site, and then they will be phosphorylated during the G1 and S phase transition. They also require certain CDK and cyclins. Remember, because CDKs are the actual proteins and cyclins, they will tell the cell whether to pass through G1 S phase or not. So once they get the signal from there, then they will start passing and then start opening the helical turn, melting the DNA strands, and form what you call a replication bubble. A replication bubble means if this is the double-stranded DNA, it slowly starts to melt in a small section first. It doesn't melt this DNA all over the place. It melts a small section, and that small section that is melted looks like a bubble. It's called the DNA bubble. This is the DNA replication bubble. And in this DNA replication bubble, if you see both this terminal, this is one point, this is one point, this is another point where the replication will pass. Let's say replication can happen in both this terminal, for example. So these two junctions, which looks something like this, if you look very carefully, it looks like this. So these structures are called as replication forks because they are looking like forks, right? They are called replication forks, and these replication forks are the machinery where all the proteins are loaded. There are other protein complexes which are unwinding these DNA strands prior in the upstream direction. Then we have other proteins, single strand binding protein, that are associated with the single strand to prevent the single strand to reanneal with themselves. And also there is the polymerase enzyme sitting and doing the job of polymerization. Okay, so that is the overview of the DNA replication in eukaryotes. From the next video, we'll be talking about the initiation, elongation and termination of eukaryotic DNA replication. Hope this video helps you, and please do watch all the videos for this series to understand properly how eukaryotic DNA replication occurs. Thank you. Like the video, subscribe to my channel to get more videos like this. All the best. Thank you.